All right, we're on our last class of biomolecules. As you can imagine, we will spend quite some time looking at nucleic acids this year when we get into protein synthesis and genetics. But when we're talking about just basic structures and being able to tell the difference, I thought it would be a good time to just get the basics in. So nucleic acids is going to be that kind of fourth class of biomolecule or macromolecule. They are also made up of carbon, hydrogen, oxygen, but in the case of nucleic acids, we're going to add in the phosphorus and nitrogen. Their main job is information storage. They carry all of the instructions that you need to build bodies and build proteins, really, and then the proteins do all the work. So they're your genetic material, DNA and RNA. They store information and they transfer information. So they give you the blueprint for building the next generation. So you have RNA, which is ribonucleic acid, and then DNA, which is deoxyribonucleic acid. So that deoxy, it's missing an oxygen in the structure. They're made up of repeating chains of monomers called nucleotides. So DNA is made up of a double strand of nucleotides, whereas RNA is a single strand of nucleotides. So here, let's take a closer look at those nucleotides. Somebody once explained it as looking at the up house. So you've got your sugar house with your phosphate balloon and your nitrogen base mailbox. If that helps you remember the structure, use it. So there's three main parts. You've got the nitrogen base, and this is going to actually differ, the part that differs between all of the nucleotides, and it's a carbon ring with nitrogen in it. A pentose sugar, so a 5-carbon sugar, so that could be ribose or deoxyribose, depending on what kind of nucleotide you're dealing with. And then a phosphate group. So those are the three basic parts of your nucleotide. And then they come together in a long chain. So again, your nitrogen base, that can be an A, T, G, C, or U. Those are the ones that differ. That kind of is where the information part is stored. You have your pentose sugar, again, ribose or deoxyribose, and then your phosphate group, which is. So these, if you're looking at the structural formula, you're going to notice that you have a sugar attached to some sort of base attached to a phosphate group. So it should be pretty easy to distinguish these from, say, a carbohydrate or a lipid or a protein. There's different classes or types of nucleotides. You have your purines and you have your pyrimidines. Those are your different nitrogen bases. So you have A, T, G, C, and then U, which is found only in RNA. T is found only in DNA. The purines are your A and G, and you'll notice those have a double chain or double ring, pardon. And then your pyrimidines are a single ring. And if you remember your base pairing rules, you always have a purine base pairing with a pyrimidine. And that actually maintains the distance, interestingly. So your cysteine, thymine, or cytosine, thymine, and uracil. So again, if you look at the structure here, you have your nucleotide on one side bound to your nucleotide on the other side, and the inside, the bases, those nitrogen bases, are held together by hydrogen bonds. So you can see those here. The DNA is going to be a double helix, so you have hydrogen bonds holding those together. And you also have, so let me go back, covalent bonds holding your nucleotides, that kind of sugar phosphate backbone, is held together by covalent bonds. So this becomes important because if you'll remember, those hydrogen bonds are relatively weak bonds, and so you can open up the molecule to read it or copy it, and then it gets closed back up again, but you're not going to break those bonds between your sugar phosphate kind of backbone as easily. I also just want to point out that these are running anti-parallel to each other. So you'll see that one side is running in one direction, the other side is running in the other direction. We say those are anti-parallel. Those fun facts. Copying DNA is called replication. And this is interesting too, because we say those two strands are complementary. So if you have a G on one side, you have a C on the other side. If you have one, you can build the other, which also means if you have one, you can rebuild the whole thing. Another really kind of interesting note, the ratio of A's to T's or G's to C's actually contributes to stability. So more GCs, there's 3 hydrogen bonds holding those together, means that it's more stable, which means it takes a higher temperature to separate those strands. They tend to be found in more like higher temperature organisms. Interestingly, we don't really know why, but parasites tend to have more A's and T's in their DNA. Some interesting kind of asides. Another interesting thing to note when we talk about nucleic acids is that ATP, that energy molecule, is actually an adenine, but instead of having one phosphate group, it has 3 phosphate groups. So this ATP actually falls into that category of nucleic acids. It's a modified nucleotide. So AMP is your adenine. It's a monophosphate plus 2 phosphate groups. You get your ATP, adenosine triphosphate. All right, quick review. Carbohydrates, made up of long chains of monosaccharides. They function for energy, raw materials, energy storage, structural components. Examples would be glucose, starch, cellulose, glycogen. So here, again, it's that dehydration synthesis is going to be used to connect those 2 sugars together. They're made up of carbon, hydrogen, and oxygen. In the monosaccharides, it's a 1 to 2 to 1 ratio. If you're looking at a disaccharide or a polysaccharide, it won't be exactly 1 to 2 to 1, but it'll be relatively close. Lipids, these are also made up of just hydrogen, carbon, oxygen, but they're going to consist of mostly hydrogen and carbon, and you'll find these kind of long chains, particularly if you're looking at your fats. They're used for energy storage. They're found in membranes, and hormones. Proteins, their monomers are going to be the amino acids. They will also be put together via dehydration synthesis or condensation reactions. They have a ton of different functions, but we're also, if you're looking at structure, they're made up of hydrogen, carbon, oxygen, but you also have that nitrogen coming into play. So you get this nitrogen, carbon, carbon, nitrogen, carbon, carbon backbone, if you're looking at the structure here. And then you have varying side groups. So you might get some sulfur in there, or it might just be carbon and hydrogen, or just hydrogen, or carbon, hydrogen, oxygen. But the main kind of identifying molecular structure is that NCC backbone. And then your nucleic acids are built from long chains of nucleotides, super important for information storage, your DNA and RNA. They are made up of your sugar, phosphate, and nitrogen base. That's what makes up your nucleotide, and they form those chains with your sugar phosphate backbone and then the nitrogen bases kind of sticking out. So those are our 4 main classes of biomolecule. Let's do a little practice at identifying them. So this guy should look familiar. Remember that anywhere those corners connect is going to actually be a carbon, and that those carbons should all have all of their valences. Their valence should be full. So if you don't see the hydrogen filled in, that you can assume that it's also there too. So this is going to be C6H12O6. So that's your glucose molecule. That would be, 1, 2, 3, 4, 5. So I lied. 1, 2, 3, 4, 5 carbons. So it's C5. I should probably not just jump ahead. And now let's count our hydrogens. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And then 1, 2, 3, 4, 5. So it is not glucose, but it still has that 1 to 2 to 1 ratio of carbon, hydrogen, oxygen. And so this would definitely be a carbohydrate. Okay, what about this guy? Here I see 5 carbon sugar, I see a phosphate, and I see these nitrogen ring structures. So that looks a lot like my up house. So that's going to be my nucleotide. So that would be a nucleic acid. So this one's a little bit trickier. I don't see any nitrogen. I only see carbon, hydrogen, oxygen. But I only see 1, 2, just 2 oxygens and a whole bunch of carbon and hydrogen. So this is likely a lipid. This is actually going to be a steroid molecule, but it is a lipid. All right, what about this one? Well, I see those zigzags, and each of those is going to be a carbon. And so I've only got 1, 2, 3, 4, 5, 6 oxygens, and this is a whole bunch of carbon, and then the attached hydrogens. So it's a lot of hydrocarbon tails, so that's got to be a lipid. And interestingly, I've got this double bond over here, so this one would be an unsaturated fatty acid. This would be an example of a saturated fatty acid. Okay, what's this one? So I've got nitrogen, so it can't be a lipid, it can't be a carbohydrate. I don't see any phosphate group. Let me see. So I have nitrogen. This and this are carbons. Nitrogen, carbon, carbon. So remember, we're looking at that nitrogen, carbon, carbon backbone. This has got to be a protein, and it's got 1 amino acid, 2 amino acids. All right, this one is a little bit tricky because I do have a phosphorus in there. I've got that phosphate, but I don't have a sugar and I don't have any nitrogen. I do have this long fatty acid tail. I've got 2 of them. So this is going to be a lipid. That's a phospholipid. So those are in your cell membranes. So it just takes a little bit of practice. If you didn't get it right away, don't worry. If you have any questions, be sure to ask me during our class time. Thanks.